Chapter 3, Topic 3.4, Eukaryotic Cell Structure. The topic objectives are: describe the composition, function, and importance of eukaryotic glycocalyxes, compare and contrast prokaryotic and eukaryotic cell walls and cytoplasmic membranes, contrast exocytosis and endocytosis, describe the role of pseudopodia in eukaryotic cells, compare and contrast the cytoplasm of prokaryotes and eukaryotes, identify non-membranous and membranous organelles, compare and contrast the structure and function of prokaryotic and eukaryotic flagella, describe the structure and function of cilia, compare and contrast eukaryotic cilia and flagella, describe the structure and function of ribosomes, cytoskeletons, and centrioles, compare and contrast the ribosomes of prokaryotes and eukaryotes, list and describe the three filaments of a eukaryotic cytoskeleton, describe the function of each of the following membranous organelles: nucleus, Golgi body, lysosome, peroxisome, vesicle, vacuole, mitochondrion, and chloroplast. Label the structures associated with each of these membranous organelles. Describe the endosymbiotic theory of the origin of mitochondria and chloroplasts and eukaryotic cells, and list evidence of the endosymbiotic theory. Now, in this topic, I'm not going to be covering all of the topic objectives, but I'm hitting most of them, and this will give you a good start. Now for the overview. We will be talking about external structures such as glycocalyces, cell walls, and cytoplasmic membranes, flagella, cytosol, ribosomes, cytoskeleton, centrioles and centrosome, nucleus, Golgi, vesicles, mitochondria, and chloroplasts. Now you'll notice in this overview that the distribution of structures in eukaryotes is flipped from that of prokaryotes. There are more interesting structures found on the inside of a eukaryotic cell versus on the outside. Well, there may be lots of interesting structures inside of prokaryotes, we just haven't found them yet, and hence we don't have to study them in this class. Like prokaryotes, many eukaryotic cells have a sticky glycocalyx, which is, once again like the prokaryotes, composed of polysaccharides. This is especially important if the cell doesn't have a cell wall, like animal cells and many of the protozoa. If you've taken A&P, you will have encountered the glycocalyx of animal cells as the extracellular matrix, which helps hold animal cells together, since all animals are multicellular. It also aids in communication between the cells. In protozoa, it helps prevent the cell from drying out, and it aids in sticking to surfaces in other cells. Now, if a eukaryotic cell has a cell wall, like plants, algae, and fungi, they don't need a glycocalyx and they don't have one. Well, some yeasts have capsules, but the rule is that you either have a cell wall or a glycocalyx. Cell walls provide protection to the cell like they do in prokaryotes. Like prokaryotes, they provide shape and support against osmotic pressure. They can be composed of many different molecules. Usually it's a polysaccharide such as cellulose, chitin, agar, or the like, but it is never peptidoglycan. That molecule is unique to the bacterial domain. On to the cytoplasmic membrane. In eukaryotes, the setup is pretty much the same as in bacteria. A phospholipid bilayer with integral and peripheral proteins. Transmembrane integral proteins are involved in both passive and active transport. In fact, the only types of transport that we talked about for prokaryotes that don't apply to eukaryotes is group translocation. Only bacteria do that. But eukaryotes don't have a cell wall that have two forms of active transport that bacteria, with their cell walls, are not capable of. Those are phagocytosis and pinocytosis. Phagocytosis is Latin for eating cell, or as we would say it in English, cell eating. The pino of pinocytosis is drinking, so pinocytosis is cell drinking. Let's go over phagocytosis first. Phagocytosis starts with the cell becoming aware of a large food source, like this amoeba attaching to this bit of algae. An extension of the cell, called a pseudopod or false foot, wraps around the food item. Eventually, the cytoplasmic membrane pinches off so the food item is inside the cell and enclosed by a membrane. We call this a vesicle. The cell can then pump the vesicle full of digestive enzymes, killing the algae and extracting its nutrients. Pinocytosis happens about the same way, with the following differences. Little bits of membrane are pinched off randomly, bringing in nutrient-rich solutions from the outside. See? Solid equals eating, liquid equals drinking. Well, enough with the external structures. Let's move on to internal structures. First on the list are flagella and cilia. Wait, you say, aren't flagella an external structure? Why, yes, I say, they are external in prokaryotes, but eukaryotic flagella are covered by the cytoplasmic membrane, as seen here in this diagram, which makes them an internal structure. See, everything inside the membrane is, well, inside the cell and considered an internal structure. Flagella and cilia have the same basic structure. The difference is that cilia are shorter than flagella, and if cells have cilia, they generally have a lot of them. So let's look at the structure. Inside the membrane, flagella and cilia are composed of bundles of microtubules that are connected by proteins. These bundles attach to a basal body, which looks about the same, but there are 9 bundles of 3 microtubules with no bundle in the middle, whereas the flagella and cilia have bundles of 2 with another bundle in the center. The flagella or cilia move when connecting proteins are contracted. That pulls the whole thing in that direction. This gives them a wave-like or whip-like motion, which is different from the prokaryotic motion. How? I've got a video clip showing both kinds. Personally, I get a kick out of the salmonella. They're so cute. Enjoy. Okay, I experienced technical difficulties inserting those clips, so you will be seeing them separately. Hopefully that'll work. Eukaryotic cytosol, same as the prokaryotic cytosol, the liquid portion of the cytoplasm. Ribosomes, we already talked about. 80S versus the prokaryotic 70S, both synthesized proteins. Now, here's something that you won't see in prokaryotes. Centrioles. Centrioles look like basal bodies. Animal cells and some fungal cells have 2 of them, arranged at right angles to each other like you see here. They are found in the region of the cytoplasm known as the centrosome. The centrosome is vital to cell division because the mitotic spindle develops from there during mitosis and meiosis. Plants, algae, and most fungi lack centrioles but have a centrosome. Okay, next up are the membrane-bound organelles that aren't flagella or cilia. These organelles are different from flagella and cilia in that they all connect to each other. As we talk about their structure and function, you'll see how. The nucleus, shown here, is often the largest organelle in the eukaryotic cell. It encloses the cell's chromosomes. Eukaryotes have linear chromosomes and usually have more than 1 chromosome. They may or may not be diploid. The chromosomes are in the nucleoplasm, may contain 1 or more nucleoli, which are areas where the chromosomes are unwound into chromatin, which is actively being transcribed. The nucleus has a double membrane pierced by nuclear pores, protein complexes that allow molecules to come in and out of the nucleus. Connected to the outer nuclear membrane by the pores is the endoplasmic reticulum, or ER. What is shown in this diagram is the rough ER, where ribosomes are embedded in the membrane of the ER. What happens is mRNAs from the nucleus come out of the nuclear pores and are transcribed in the rough ER. What is not shown is the smooth ER. The smooth ER is also embedded with enzymes, but they are too small to be seen with a light microscope. These enzymes are involved with carbohydrate synthesis, lipid synthesis, and in the mammalian liver, breakdown of drugs, alcohol, and other molecules. In both types of ER, little blebs of membrane are pinched off to form vesicles. These vesicles contain brand new proteins, lipids, or carbohydrates. They aren't finished yet, though, usually. The vesicles are transported to the next membranous organelle in the cell, the Golgi body. So here we've got vesicles coming from the ER and they fuse with the membrane of the Golgi body. Within the Golgi, the macromolecules are modified. A protein may be snipped, a carbohydrate may be added, a lipid and a carbohydrate may be fused. The possibilities are endless. When the Golgi is done, more vesicles pinch off the other side. These vesicles are transported all over the cell, to wherever the finished macromolecules are needed. Some vesicles fuse with the cytoplasmic membrane, carrying integral proteins and the like. Some vesicles, called secretory vesicles, carry macromolecules that are intended for export from the cell. An example in humans would be insulin being secreted from pancreatic cells. Some vesicles, called lysosomes, contain hydrolytic enzymes, which are used to break down the contents of phagocytic vesicles, or phagosomes. They also break down worn-out organelles, enzymes, and the like so that their components can be reused. Lysosomes can also break open during programmed cell death, or apoptosis, destroying the cell from the inside. Peroxisomes are specialized vesicles, like lysosomes, but they come straight from the ER. They degrade the dangerous forms of oxygen that are generated during aerobic respiration. We'll talk more about them in later chapters. The vacuole is another term for vesicle, but it's usually reserved for the large structure in plants and some algae cells. It's so big that the cytoplasm is a thin ring around the edge of the cell, next to the cytoplasmic membrane in the cell wall. It is used in controlling water pressure in the cell, for storing nutrients, and for storing waste until they can be exported. Onto the membrane-bound organelles that are not directly connected to the endomembrane system, mitochondria and chloroplasts. Both have double membranes, like the nucleus. They both have extensive folds in the inner membranes. In mitochondria, these folds are called cristae. Enzyme complexes involved in aerobic respiration are embedded in the cristae and are also found in the matrix or inside of the mitochondria. Chloroplasts, which are the sites of photosynthesis in plants, algae, and some protozoa, have thylakoids that are stacked into granum. Enzymes required for photosynthesis are embedded in the thylakoids and are found in the inner space called the stroma. Sounds like the mitochondria, huh? Both have 70S ribosomes, and they have their own circular chromosomes which contain genes specific to photosynthesis and aerobic respiration, and they divide on their own, independently of the rest of the cell. Sounds like prokaryotes, doesn't it? In fact, genetic research has found that the DNA in mitochondria is very close in sequence to some forms of aerobic, free-living bacteria. The DNA in chloroplasts is very close in sequence to cyanobacteria, which are photosynthetic. So how did that happen? The current theory is that early eukaryotes engulfed the ancestors of the mitochondria. Instead of being eaten, they survived and eventually lost the ability to survive on their own. The eukaryotic cell took care of them, and the mitochondria produced most of the energy for the cell. Later on, some eukaryotic cells engulfed the ancestors of chloroplasts. The same process took place, to the point where plants, algae, and some protozoa are able to use light energy to synthesize sugars from CO2 because of their chloroplast friends. Do remember, though, that the endosymbiotic event that resulted in chloroplasts took place after the endosymbiotic event that resulted in mitochondria. How do we know that? Because all eukaryotes have mitochondria, or organelles that used to be mitochondria. Not all eukaryotes have chloroplasts, so that had to have been a later event. Well, that's it for this topic. I have some summary slides. This one shows an animal cell. It would be good to practice labeling this figure with the names of the structures, both external and internal, and with the functions of the structures. I have included a table that summarizes the various structures, lists their functions, and indicates if they are in prokaryotes, eukaryotes, or both. And here are the reminders of the learning objectives for this topic.